Hello everyone. This is meant to be a quick review here for you. Kind of just a general overview for what you need to know about cell division and mitosis and meiosis. It will not give you everything you need to know, but it's a pretty good overview. So if you could pull out your condensed notes that I've given you, I'm just going to kind of go through it bit by bit. Um, so first thing you need to know is what's the overall purpose of cell division. Um, Generally speaking, though, the overall purpose, as you can see up there at the top, when cells need to reproduce, when cells need to grow, and when cells need to repair themselves. So, generally speaking, those are the three main things. We have our core concepts listed below here. Um, all organisms consist of cells and arise from pre-existing cells. Mitosis is when all regular body cells are created. Meiosis is when we produce sex cells that are used for sexual reproduction. So if there's one little takeaway that you could get, one single takeaway is meiosis makes sex cells or gametes, mitosis, everything else. So if you get a little tripped up on that, again, just remember meiosis, sex cells, mitosis, literally every other cell out there. We also talk a little bit about the cell cycle, which is actually should, in my opinion, be called the life cycle of the cell. So really, the cell cycle, the actual division part, is only a very tiny bit of the cell's life cycle. And also, what can happen if cells are mitotic divisions or cell division is not regulated. We know it can happen when cells divide too much. We know that leads to cancer, so we'll talk about the checkpoints very quickly. Um, I have here just some basic pieces of terminology for you should be familiar with: diploid, haploid, things like that. Chromosome structure, it is important to know. The center of the chromosome in this general region is your centromere, created by little kinetochores. I'm going to scroll down a little bit further. This is just showing you generally how chromosomes are supercoiled or wrapped. They're actually wrapped around little histones, the little proteins that the DNA can wrap up on. Um, so if we look at the cell cycle very quickly, again, this is just meant to be a quick, brief overview here. We start the cell's life cycle in G1 or first gap. I also like to call it growth 1 phase. In growth 1 phase, the cell is literally just getting a little bit larger. It's maturing just as we mature in our lifetime from children to about up to adulthood. It's just growing and maturing. There will be a checkpoint from G1 going into S when we talk about regulation. But once it does enter into S phase, also part of its regular life, in interphase, it's called S phase, which stands for synthesis, and we're synthesizing new DNA. It's important to know we have to double the DNA before we divide the cell. If we don't double the DNA before we divide the cell, then each cell will only have half the amount. And the overall purpose of mitosis is to produce two identical cells. So we want to duplicate or clone the DNA. In gap 2 or growth phase 2, the cell will prepare to divide. And there will be one more checkpoint right before it goes into M phase, which is mitosis. But right before that, we're going to hit a checkpoint, and it will then go into mitosis, which you know as prophase, metaphase, anaphase, telophase. So notice here that interphase makes up most of this life of the cell. Only this little tiny bit here is where it actually divides, which is mitosis. So you need to keep that in mind. If we divide too much, cancer cells basically skip this step. They skip interphase. They jump straight into mitosis. We don't want that. So mitosis in a nutshell, the stages of mitosis can be broken down into 6 stages. And actually, I should probably delete this section here, which I shall do. Interphase is not considered part of mitosis because interphase the cell is not dividing; it's prepping to divide, um, and the cell is simply doing its job. So the actual part of mitosis is what I call PMAT, PMAT, prophase, metaphase, anaphase, telophase. I would also like to scroll up here and also show you one other thing before I talk about mitosis. If the cell is never going to divide again, it's like, let's take a nerve cell, for example, a nerve cell out of your spinal cord. It will actually go into G0, which is what we call a resting state. So once your nerve cells are created, they go into a resting state, which we call growth 0, and they will never go back into the cell's normal division and things like that. All right, so back to mitosis. We have our DNA duplicated. We're ready to divide. We're ready to create literally an identical clone of the cell. That's the overall goal. 2 sister cells or daughter cells. We start the first step, and actually this animation isn't that great. I'll have to redraw this at a later date, but in prophase 1, the nucleus, which you see surrounding the DNA here, begins to dissolve. That's why there's little breaks in it. You can see the centrioles up top here. And actually, what also begins to happen is that the spindle fibers will begin to form as well, which you can't really see here. Also, the X chromosomes will become visible. So chromosomes condense, become visible. The mitotic spindle forms, which only occurs in animal cells. There's a kind of a half step, a prometaphase. This is when the cell or the spindle fibers start to push these chromosomes towards the middle of the cell. So you can't really see the X's here. This animation is more or less showing the really beginning starts of prophase. But prometaphase, centrioles and the spindle fibers, these little extendo fibers, begin to push the chromosomes towards the middle of the cell, which we call the metaphase plate. It's an imaginary plane, sort of like the equator. We cannot see the equator, but we know it's there on the Earth. Here you can see the metaphase plate on the cell. The chromosomes line up end to end. In metaphase, meta means middle. You can also see the image here in the electron. Oh, I'm not sorry, that's not an electron microscope. That is actually a compound light microscope. You can see this under a standard microscope. Anaphase, it's kind of logical what would happen if we're trying to pull these apart. The spindles grab a hold of the centromere or the kinetochore. They start to pull them to opposite ends of the cell. So now you have half the chromosomes on one side of the cell, half on the other. You can see the real life picture here to the right. Then in telophase, some people say telophase, the nucleus begins to reform. The spindle fibers are now gone. In animal cells, a cleavage furrow is formed, which is kind of this little dip. The membrane actually pinches off. There's a little band that will actually pinch this off. And the cell will completely divide when the cytoplasm fully pulls apart. We call that cytokinesis. Cyto, the prefix literally refers to cytoplasm. So when the cell fully pinches apart, we call that cytokinesis. A lot of books will combine telophase and cytokinesis, so it sounds confusing. Just know they're kind of similar. Telophase, spindle fibers dissolve, the nucleus reforms, the chromosomes unwind. Cytokinesis is the fully pinching off of the cell. I already said that only animal cells form a cleavage furrow. When they pinch apart, literally the cell membrane squeezes like a bubble, pinches apart. In plant cells, they do not do that. Plant cells, the cell wall is so strong, they cannot pinch. And actually a cell plate will actually form right across the middle, and then it will just fully grow apart. And really that's all you need to know about mitosis. So 2 daughter cells, completely identical. So if you start with a diploid cell of 46 chromosomes, you want to end with a cell with 46 chromosomes. You start with a cell that has 200 chromosomes, you want to end with a cell that has 200 chromosomes. identical cells. I'm going ahead and go to skip to meiosis, and then I'll come back to regulation. Meiosis, very similar, except for a few steps. Step one that is different is in prophase. So first of all, you need to know there are 2 rounds of division in meiosis. And that's because we have to have the cell twice. So first thing you need to know, we already know that you get half of your DNA from mom, half from dad. Most of us know that. Here's where it changes it. Note, when you saw in mitosis, the X chromosomes formed, they just formed and that was it. In meiosis, not true. Meiosis, we're trying to produce half of the DNA. So one of the big things that has to happen, first of all, each chromosome has to pair up with its matching pair. The one that came from mom, the one that came from dad, that matches for those same genetic traits. We call those homologous chromosomes. Prefix homo means the same. So in prophase, not only do the X's form, but they pair up with their matching partners, their homologous partners. Also, one thing I'd like you to notice here, you see when they pair up, it's like their arms are touching right there. When their arms touch, sometimes they will, sometimes they won't, but they'll actually exchange the DNA on those arms. We call that crossing over. And crossing over is the first big thing that leads to each sex cell making a unique combination of the DNA. So basically every sperm that's created, every egg that's created will be entirely different from every other egg and sperm. So there are a few other things that are going to lead into that diversity, but that's one of them. Okay, so we got crossing over and the pairing up. That's step one. And there's a little picture of 2 chromosomes crossing over here. You get a unique combo of them. We also call this formation of the pairs here. We call this a tetrad. You have 4 single-stranded or 4 chromatids pairing up. Tetra means 4. So we call this a tetrad. All right. Second big difference, remember in, let me scroll back up here. This is page 14. Go back to that. This is mitosis. You see here, the chromosomes line up in a straight line, straight across. So when you split it apart, you're going to get the same amount on each side as the beginning cell. So if this cell started with 4 chromosomes, you have 4 on this side, 4 on this side. If I look back here in meiosis, now they're not lining up end to end. They're lining up in their pairs. That is very different. So if you started with 4 now, when they pull apart, look what happened. They pull apart in anaphase. Look at this. Now each cell only has 2. Now we're down to half the amount. Half the amount number means haploid. But here's the thing, when it actually gives half the amount, we can still cut it one more time. Each X that we see in a cell still has double the amount of DNA required for a gamete or for an egg or a sperm. So just to make this a little more efficient, we're still going to cut this in half one more time. There's no crossing over in prophase 2. The chromosomes are getting ready to pull apart again. They line up in metaphase, not in pairs, because we already pulled them apart from their matching pair. There's no one to pair with. Their pair is already in the other cell. So they line up in a straight line. Again, the initial cell had 4 chromosomes. These have 2. We're going to pull them apart one more time. Now we have 4 cells here, each with half the original amount. These are your sex cells. In spermatogenesis and the creation of sperm cells, you will create 4 sperm cells. In oogenesis, the formation of egg, 1 egg will absorb all the cytoplasm and the organelles from the other 3, and they'll become polar bodies and dissolve, and only 1 mature ovum will form. This ensures that you form 1 perfect egg each time. I'm going to continue this lecture because I'm about to run out of time here. I'll pick up next time with cell regulation and how that leads to cancer, and that'll wrap up the unit. Have a great day.